Bismillahirrahmanirrahim. Assalamu alaikum, my very dear students. Your teacher Sayyida Sara is here. Again, we are doing one more technique right now, and that is the technique of staining, which is very important in the cell biology. And staining is the technique which is also called as dyeing. To dye any object or to dye any cell is the technique called as staining. Basic purpose of staining is to enhance the visualization. Certain cells or most of the cells, like bacterial cells or the cells of the body of any living organisms, are not colored. So to enhance their visualization, cells are dyed or cells are stained. So we not only can observe cells fully, but also cellular components which are called as organelles can easily be observed after the staining techniques. Staining purpose is also to differentiate between the live and dead cells. There are certain parts or there are certain cells which are the living cells along with the dead cells. So dead cells can be separated out or from the living cells in a sample medium. So staining technique will help us. We can take example of bacteria. That bacteria are colorless, and to study structure and its shape, it is necessary to make its visibility under the microscope by the technique of staining. Likewise, plant cells are also observed well under the microscope after staining. So after staining, cells are observed or examined well under the microscope. So that's why we say that staining is a technique which is used to enhance the visualization of any cell or component of the cell. Dear students, there are certain types of stains which are used for the living cells. For example, one stain is called as vital stain. Vital stain is the first stain which is given to the cell. And then a second kind of dye or stain can be used, and that is called as counter stain. One of the good property or the feature of the such kind of stain is that these are harmless, these are non-toxic, these are used in very low concentration, and these are used within the living cells in order to highlight them, and this will increase the resolution of the microscope, and we can easily clearly observe them under the microscope. The type of the stains which are taken as vital stains are methylene blue and neutral red. There is also a long list of certain stains, but I have mentioned only two. Similarly, as a counter stain, saffronin is used. Now girls, on the basis of the type of the or nature of the stain, we divide the type into three basic types. First is the basic stain, second is the acidic stains, and third category is the negative stains. First basic stain category includes same methylene blue, crystal violet dye, and saffronin. These three names I have included. Similarly, for acidic stains, eosin, which is also called as eosin solution, rose bengal, and Congo red are used, which are in the category of acidic stains. Third category is negative stains. Negative stains include India ink and nigrosine. That is nigrosine. Dear students, there are 5 types of staining as well. First is called as simple staining, and single dye is used in this simple staining. Second kind of staining is called as differential staining. And this differential staining, from name is clear, it is to differentiate or to separate two organisms from each other, or two parts can also be separated with the help of differential staining, a kind of which is gram staining. So the third type is gram staining. In 1884, a Danish physician, Hans Christian Gram, introduced the technique whilst he was working with the staining of bacteria. And he introduced the techniques. There are certain steps of those techniques. The first step is the use of primary stain. Mostly primary stain used is the crystal violet stain. In the second step, he introduced iodine solution. He used iodine solution, and this treatment was taken as mordant. Mordant here means that will enhance the coloring of the, or the dye of that stain. Means that dye or that stain will be easily taken up by the bacteria. Third is decolorization step, and this decolorization is normally done with ethanol. Acetone can be used to decolorize the stain. Then fourth step is gram-positive retained the violet dye. So gram-positive bacteria retain the violet dye, so that's why those bacteria are known as gram-positive bacteria. Girls, the basic difference between the gram-positive and gram-negative bacteria is lying within the structure and composition of their cell walls. Girls, as we know that basic difference lies in the difference in the cell wall, gram-positive bacteria retain the crystal violet dye, but gram-negative bacteria did not. Actually, gram-positive bacteria cell wall was thick, but their cell wall was made up of very simple components. That's why that retained the crystal violet dye. But cell wall of gram-negative bacteria did not retain the crystal violet dye because it was thin but made up of quite complex components. Another step, what Sir Hans did, he took the smear, and that smear is counterstained, and that is counterstained with another dye which is known as saffronin, which will impart the pink or red color, not red but bit pink color. So gram-negative bacteria were stained with pink color and they retained their pink dye. Another technique of staining is acid-fast staining, and this acid-fast staining is used for mycobacterium. And another technique is endospore staining. In unfavorable conditions, bacteria start to produce endospore in order to prevent itself from dying. Dying means here death. So in order to prevent it from unfavorable condition, bacteria produce endospores, and this is a technique which is called as endospore staining. This endospore staining is used for bacterial endospores in order to stain them and to observe them keenly or to observe them deeply. So girls, these are all the 5 basic types of staining.